Thank you. Well, good morning, and thank you very much for the invitation for me to come and share some of the works that we have done in regards to looking at different technologies, particularly in this area of low volume dispensing. So one thing that we did through these studies was, certainly made possible by internal partner and collaboration. And so I've listed some of our colleagues in Dow AgroSciences that are kind of going through a lot of these day in, day out type workflow, and also looking at aspects of how can we further improve and optimize, especially in the area of miniaturizing volumes of samples. So before I kind of dig deeper into the type of studies that we did and what were some of the key learning and findings, I'd like to kind of give a brief overview of who we are and what we do at Dow AgroSciences. So we began in 1955 as an agricultural unit of Dow Chemical Company. So essentially what happened after that was in the 89 kind of an area, Dow Chemical entered a joint venture with the Plant Sciences Division of the Elanco business of Eli Lilly to form what they call Dow Elanco. And they built their headquarters in Indianapolis, Indiana, which we are still located today. So around 1997, Dow Chemical acquired 100% ownership of the joint venture and renamed us to Dow AgroSciences. And so today we are a wholly owned subsidiary of the Dow Chemical Company, and one of the exciting things that are happening with our company is the merger that we are currently undergoing with DuPont. So that's certainly a very exciting thing for us to be part of as we build out the agricultural business. So what we do is really focused on the agricultural aspect of the industry. And our key focus is essentially the farmers. And we are trying to develop solutions to help kind of meet the needs, the growing needs of the farming industry. And we do that through several key crop sciences areas. So we're involved in plant genetics and seeds, crop protection, urban pest management, vegetation management, range and pasture, plant nutrient management, as well as post-harvest technologies. So you can see that we are quite diverse and we try to cover the entire spectrum in this area. So one of the key initiatives within the R&D automation group was really to look at developing new capabilities and platform to help our researcher. So one of the topics that we are very passionate about is in the area of low volume dispensing. And we wanted to understand as well as evaluate, what are the options that are available out there in the market and how they could potentially apply to our internal workflows. So what we did about 2 summers ago was actually put together an intern project and as well as interacting with our internal partners in the different areas. And we really want to understand what are some of the capabilities that are available and also assessing the type of workflow that we are engaging in, particularly in the areas like next generation sequencing, in the traits assessment that are more high throughput methodology, and most recently kind of looking at the system biology area in the high throughput DNA assembly realm. And so we quickly kind of assessed about what are the key factors that we need to kind of keep in mind as we look at the different available technologies coupled with our internal workflows. So as I've mentioned, we are always striving to understand what we can put in place to support our high throughput screening platform. Cost is another factor that all the industries are going after. How can we reduce our operation cost? And how do we maximize efficiency in terms of processes, people resources, and even enhancing productivity? So those were quite our key drivers in helping us to line up a project plan when we're doing this assessment. So one other aspect of this whole study was to kind of identify which partners are we, trying to bring together to do these studies. At the same time, having this discussion with our internal partners to understand what are some of the key criteria that they are looking for before they are willing to adopt new technologies into their workflow. So key things here is obviously, when it comes to liquid dispensing, how accurate it is, how precise it is. That's very critical and crucial to us. Performance, we need to have consistency in the way the system perform, as well as in helping us with our workflow. And what are the areas that are pretty much what the industries are going after? As I've mentioned, in the NGS area, in the next generation sequencing. That is a capability and a tool, an important tool for us at the Dow AgroSciences, because we use that to do a lot of validation of constructs that we have put together that eventually becomes the next product for our seeds and traits platform. At the same time also, we use the sequencing to look at new novel genes that we could incorporate into our cash crops, such as corn, soybeans, canola, and have them be more insect resistant, maybe even have higher nutrition value to help kind of feed into this product portfolio of ours. And why this is so important is because the way that our business is set up, we have these two platforms, the seeds and traits and the crop protection. And both really demands that whole high throughput screening capability, low operation cost, as well as efficiency in productivity and workflow. And so when we were looking at what to investigate and what to kind of incorporate into this, we were really kind of leveraging studies and findings that have been already put in place in the pharmaceutical industry as well as in the academic world. So what we did 2 summers ago was that we engaged in collaboration and partnership with key external vendors. And so we partnered with Labcyte to really understand what the ecosystem capabilities are from a non-contact dispensing approach. We also partnered with TTP LabTech to look at their Mosquito system. As had been mentioned before, they have the capability of assessing certain workflows that involves like bead cleanup, where acoustic technology may not be the most appropriate system to utilize. And so we enter a collaboration with them to kind of help us form this structure a little bit more. And I have to say that both partners have been really, really great in helping us when we were setting this up. They not only provide, technical support, but also the scientific aspect of it. So it has been a very good partnership, I have to say, and a continuation of partnership even up until now. So what I'm going to talk about is some of the workflows that we had looked into when it comes to assessing these different technologies in this area. So one of the workflows that we look into more in depth is the Nextera and the Nextera XT. And how we use this protocol is really to prepare DNA from smaller genomes, such as bacteria, for sequencing validation. And this workflow is typically geared towards our seeds and traits kind of crop area. And where it becomes very valuable is when we introduce new traits into, let's say, corn. We need to quickly screen at the lab level, and I'm talking about millions of samples, very rapidly, so that we can get the data out back to our field scientists to make the appropriate decision of what plots to keep and what plots to quickly remove because of the sensitivity of trying to, timeline sensitivity to get the product's advancement out the door. And between the two assays that we have put in place, we typically go towards the Nextera XT because it's been most cost effective for us. And so I've kind of highlight, the key steps that are involved in our internal protocols in terms of both Nextera and Nextera XT. And so what we were essentially focusing in this study was the segmentation of the genomic DNA component. And what I meant by that is really looking at where the DNA is tagged and fragmented by the Nextera transposome, and specifically kind of understanding how can we miniaturize the steps on the DNA as well as the segmentation enzyme and buffer addition to this workflow. We're also still working on understanding how can we further optimize these other steps, particularly in the normalization and the pooling component for sequencing. So we're continuing kind of using our existing ecosystem to look at how can we miniaturize that workflow and how can we optimize it further. At the same time, we're also thinking about what if we introduce the Mosquito system back into this workflow to kind of really have that synergy effect to help us to streamline the entire thing. So in this study, what we were focusing on is really, can we reduce the volume of the protocol that was being developed? And we actually utilized some relevant concepts from the multiplex preparation study that was done and published in 2015. The only kind of deviation that we did was we combined our segmentation, buffer, and reagent together prior to having it dispensed into the what we call our daughter plates. And the reason we do that is because we were trying to minimize any variability so that we can control it a little bit more effectively. We're also looking at the different DNA concentration level here. And what we chose was 1.7 nanogram per microliter and 0.2 nanogram per microliter. And the rationale behind these two DNA concentration is really based on the existing data sets that we have been collecting to try to kind of push the limit a little bit, just to understand, how far can we go until we disrupt, the efficiency of the data that we have been seeing. And the other part was, obviously the most important thing was looking at volume. And so what we were assessing was what we call a full volume based on what's been developed in the protocol versus half volume assessment. So we literally reduced the volume in half of the total setup. So these are some of the results that we obtained from the studies. So looking at the two different DNA concentrations at both full and half volumes, we quickly found that the 1.7 nanogram has no issues at all. It made the library that we expected it to make at both the full and the half volume, which was great news. What was a little surprising is that the 0.2 nanogram microliter DNA concentration, it made absolutely no library, regardless of what we did with the volume range. So that's kind of where we saw, oh, this is really that extreme, breaking point of don't ever go there, because you're not going to be able to qualitatively or even quantitatively assess what is going on with your workflow. We chose more of a qualitative assessment route to kind of understand our results because of time constraint. Typically what we would do is we would send it off for sequencing just to get that full understanding of what we're looking at. And so we chose this electropherograms just to kind of give us that visual qualitative assessment of whether we have the results that we intend to have or we are so far off basis. And we also did this test in both the Echo as well as the Mosquito system. What we did was we tried to standardize a little bit of the methodology, and so we used the 384 well qualified plate from Labcyte as our source plate and used that in both systems to do the dispensing and the transfer. And also, when we were doing some sort of a cost calculation to help us make that decision of what system to bring in-house and incorporate into the workflow, so we were looking at ROI. So before we made the change, the operating cost for these protocols were essentially at about $27 per sample. So it was pretty costly. And so that kind of limit our ability to run this as a standard high throughput operation for us. By going through these studies in terms of reduction in volume, we actually were able to drop the cost down to $7 per sample and $3.50 per sample. So essentially this made it very cost effective and feasible for us to introduce this into our workflow as sort of a kind of high throughput screening capability for us. So that was really well received by the company. So another internal protocols workflow that we assessed what was our KASPar assay. So this is an in-house developed protocol that are used for our seeds and traits platform. What we're trying to do here is really kind of have a methodology put in place to help us select appropriate traits. Again, that was the one that I talked about, having that quick turnaround time for data collection and analysis to feed back into our field biologists so that they can make the appropriate selection of the right traits out in the field area. So just to kind of give an overview of how this process works, what we do is we cross the elite parent with the donor parent to form children, what we call progeny. And what we're trying to do is really select those right traits that occurred in this progeny, the offspring. And essentially, what we consider as a good or desirable progeny is the one that contains the gene of interest from the donor parent. You do get some mixes where you have a gene pattern of just strictly from the elite parent or strictly from the donor parent. So essentially, we want the combination of desirable traits from both to give us that progeny. And in here, we're really focusing on volume reduction of particularly the markers, internal markers that are developed by AgroSciences, as well as the assay master mix for this protocol. So what I've put down there is just sort of the series of steps that occurred for this assay, from isolating DNA, preparation, all the way through to data analysis. And so we chose the characterization plates preparation area because this is the area where we are seeing a lot of high throughput coming into place. I think about last year alone, they processed through, I think, 2 to 3 million data points in the period of what we call the busy summer months, so that we can advance the data out back into the field for them to make decision. And we have projection of actually double up the throughput needs in the coming years. And so we have to really start looking at that next generation platform setup with technologies like the Echo to help us manage that workflow. And then also one thing too is the whole dead volume reduction was very important to this internal partner. So they use the LGC Meridian dispensing system that has huge high dead volume. And because of the reagents and the markers that we're using, they're pretty expensive. And so we can't really afford to dump out what is left at the end of the run. We would like to try and conserve as much as possible. And so that was one of the key drivers that kind of got them excited, interested of what the system is potentially can help them out with in terms of cost reduction there. So we tested several working volume ranges, and particularly in replicates, because the way of their workflow structure, we can have a lot of number of samples going through the pipeline. And we use what we call a control system that they are currently using in the Meridian dispensing system as a way to kind of measure how effective these new technologies are in getting the desirable end results from them. Basically not impacting any of their end product in a negative manner. So what we did was run through the series of the test, and we did it in parallel with their control using the Meridian dispensing system. And we looked at different volume ranges, starting at 945 nanoliter, which best match with what they are currently doing in their workflow, which is at 1 microliter volume range. And we try to push the limit down and try to see where is that kind of end spectrum, right? Can we go lower than that? And so we chose to end it at 315 nanoliters. So one exciting thing that we found out was that the 315 nanoliter was not the extreme end. Essentially, we can still go further down in terms of volume reduction and not negatively impacting our end results. So that was certainly very exciting for us. And what we're doing here is our internal kind of data analysis software, where we are looking at patterns distribution. And so these series of red, blue, and green dots, they depict, something of importance to us. What we are essentially looking at or trying to get out is the green dots. These are the ones that contain the gene of interest from the donor, but has some of the characteristics of their elite parent. And here we're trying to figure out, from a pattern distribution standpoint, what volume range still meets that same standard that we have been using to do our analysis. As you can see with the ECHO system, the pattern doesn't really change a whole lot. It is very consistent to what we have been getting using our regular protocol and SOP. So that was interesting, as well as good news for us, that we could push the limit of this workflow down quite a bit. And we also looked at percent error in terms of how we would have been getting results using the traditional workflow. So how many did we actually miss? 3.7% is not much. So this is well, below the acceptable, within the acceptable range. I mean, in our traditional workflow, we missed as much as 5 to 7%, and that was still deemed okay. So to see that we only missed 3.7% using the ECHO system in this new workflow was certainly very promising and exciting for us. We also ran the same test in the Mosquito system with the same volume ranges and then also with the same data analysis. The pattern somewhat stayed the same, but what we noticed was the volume range at 788 nanoliters. You see this spread of the points. And what we quickly realized was that the configuration of our destination plate, that 384 well plate, has this very narrow wells in between. And so every time the Mosquito pipette tips come in contact, it gets bent. And so that causes us to not have that even sample distribution. And also we noticed that as you dispense your sample across the plate, you get this kind of drag effect. So we were seeing a lot of cross-contamination between wells, which is not acceptable at all. And so once we figured out and noticed what was going on with that volume range at 788 nanoliters, we basically program it to switch tips every single well at 630 nanoliter all the way down. It did solve the problem. However, it drove our operation cost way up. So you know that if you own a Mosquito, their tips are not particularly very cheap. So to change it out every single well and knowing that we are going to go through millions and millions of sample, that's not going to be very cost effective for us. But regardless, we're still able to demonstrate that you can essentially push the limit of the volume, down further, whether it be the ECHO or the Mosquito system. And here, because of the drag effect as well as the bent of those tips, we actually saw an average percent error, meaning hits that we missed, in the 5.2% range. Still acceptable, not too bad, but when you compare it with the ECHO, we certainly thought that ECHO was giving us meeting the requirements that we have set it out to meet. So we did a assay cost analysis just to kind of understand where we're at, and because we were also trying to present to the management at the end of this studies that, hey, we would like to acquire one of these systems, if possible, both. And so that's why we have to do an ROI, just to kind of paint that picture a little bit. As I've mentioned, with this particular assay, with the Mosquito system, we didn't really see a positive return of investment due to the fact that we have to change tips for every well that we're dispensing. And so we lost track of how much it cost us after a while. We said, all right, it's just negative, and it's not really worthwhile for this particular assays. But for the echo, we saw our return of investment really quickly. I mean, less than half a year, we get it back. And this was certainly a no-brainer for us, that to incorporate technology into this workflow, echo was our solution. Okay. So what are some of the key learnings that we got out of the both Nextera and the KASPar assays? The echo system demonstrated excellent consistency and flexibility between these tested internal protocols. We also saw that the echo is more accurate and precise in the liquid dispensing compared to Mosquito, particularly for our KASPar assays. However, I do want to stress that the Mosquito is comparable to the echo when it comes to certain portion of our workflow, particularly in the assay mixes. It's capable of doing that in just that one system setup. But ultimately, it did show us, the consistency is not quite there for us, and therefore not suitable for our KASPar assay, as well as the whole consumable cost with the pipette tips that we have to change out for every well. For Nextera XT, we did talk to our stakeholders because they didn't really experience a lot of major issues with Mosquito HTS system. We thought it would be a better option for them, particularly for their protocol. One thing is, I've mentioned, they're able to perform this mixed step within the wells. And so you can help decrease the hands-on time of their limited people resources. At the end of it, management told us we can only pick one system to bring in-house. And so looking at all the data that we have, as well as trying to think ahead of the other potential applications that we need to build out capabilities for, we ultimately went with the echo system because we felt that it was the most reliable, versatile for these different tested internal protocol, as well as for any future protocols that we want to develop. And also from a return of investment perspective, we felt it is way more cost-effective investment for us. So what has happened since then, based on these studies that we've done? We've gone ahead and developed an automated test bed system that incorporates the echo and several other key instrumentation to kind of really help build out that full automated workflow. And also, since then, we have expanded the ecosystem to other applications in order to develop new capabilities, which I'll talk about in my next few slides. So one of our latest kind of workflow development and capability development, partnering with some of internal key stakeholders, is in the area of the system biology, in particularly our high-throughput DNA assembly. And this is really to feed into, again, our seeds and traits platform. We are, being asked to help, shorten those production line time frame to launch product out in the market faster. And so one of the things that we can do to help shorten that timeline, it's in the upstream portion of it, particularly in the DNA assembly area, to help develop more constructs with key genes of interest with the right traits so that we can really put it into our plant models from corn all the way to canola to do the testing. So what has been happening, I would say about a year now, is we were building this whole automated and high throughput platform to help support this DNA assembly workflows. So we have a team of biologists as well as a team of engineers from my automation group to help build this thing out. And additionally, we're also looking at data management perspective. So what is happening here in this workflow is that we identify several steps that are occurring. From what we call parts and pieces, which is the start of the design piece, and identifying what are the ingredients needed to construct these end plasmids. Follow into, okay, now you have all these different ingredients or parts and pieces, how do you put them together, which we term the assembly. And that was really the focus for our capability development from a lab operation standpoint and using the ecosystem. And then it goes into what we call a quality control. That is more on the sequencing side of things. So now you have all these construct made. How do you know which one actually contained the traits and the gene of interest that you have incorporated into? And so that's where the whole data management system comes into play, as well as opportunity too for lab automation operations to help miniaturize those steps too. And down to our storage, our archiving. Because a lot of these construct and parts and pieces that it contains is novel to us. Some are made in-house, some are kind of sourced out to get those libraries built out so that we can keep using it for other projects. And the key drivers, that prompted our stakeholders to, really sit down and think about how can we optimize this workflow was based on their current protocol. Their entire workflow essentially now is taking weeks to complete. And each step in the workflow actually has several sub-steps. And there's very minimal lab automation involved in the original workflow. A lot of it is technicians and contractors sitting down in the lab, manual pipette, doing that kind of build up plate by plate. And so we said, that's kind of crazy, and you're expecting to up your throughput. How can you keep up, with just hiring more people and using manual, tools to conduct that type of processes? At the same time, we're also noticing, from a data integrity piece, there is that consistency issue, and we need to have that ability to correlate information from one processes to the next. And so automating and reducing the volume, particularly for the assembly step, would essentially be kind of the first area for us to focus on. And so where we bring in the echo is really helping us to combine all these multiple parts and pieces with the assembly enzyme to help us form the plasmid that we can put into construct for testing. So I talked a little bit about some of these different steps that are involved. And so with the very initial piece of design plasmid, we are also engaging in collaboration and partnership with an external partner that are specializing in developing tools in this area. So how do we create these libraries of parts and pieces? How do we track it? How do we manage it? And then what are some of the algorithms that you can put in place to help us put together some prospective design so that we can assemble it into plasmid, then put it into construct for analysis and testing. So that's still an ongoing work, but so far what we have seen was very powerful, and it certainly has helped reduce our cycle time in terms of trying to get all of these analysis done in a timely manner, as well as putting it into eventually our plants for testing. So what are some of the kind of aha things that we got out of this study, which is still ongoing? We found that the echo has been a very good fit for our DNA assembly workflow. Not just from the front end of things, but we see a potential kind of in our downstream processes, particularly in the sequencing. We were able to reduce our reaction volumes down to the 2 to 5 microliter range. They were actually using 20 microliter in their original protocol, which was quite costly in terms of reagents and the parts and pieces that they have to put together. We try to push the limit a little bit, but based on the current way of the workflow is being designed, there's really no way for us to further reduce the reaction volumes. But they were very happy the fact that we can bring it down from 20 all the way to 2, which was a significant improvement in how they are approaching this whole workflow. The reason that they provided was the fact that a lot of our subsequent downstream steps is highly dependent on sample volume at the microliter range. But I do believe that as soon as they are able to develop the sequencing portion of that analysis, we can try to push that volume envelope a little bit more. So it's, you know, we're still very much hopeful that we can get even lower than the whole 2 microliter range. So currently, we don't have plans to do that, but it's still on our radar. We're still trying to push the envelope. At the same time, what they have found was that, you know, they're able to optimize, you know, even at the microliter volume range on the echo very easily. Because one of my kind of stakeholders that are involved in the studies was pretty apprehended in the beginning. Because when they hear, you know, nano volume technology, they're thinking of, wow, that's really low in the volume, but we're not quite there yet. How would it be able to accommodate, you know, the type of workflow that we are working with, particularly in the very low microliter range? So he was very pleasantly surprised at the fact that Echo was able to handle that type of limit. And they share with me that their data that they have been getting using the system helped them to achieve significant throughput increased, as well as cost savings with the use of the Echo system. And most importantly for them was the reaction efficiency. They do not want to see any negative changes to that, and they did not notice anything like that at all with the volume reduction. And so that was certainly very nice to hear, and they were able to use this and convince their management to really do a shift and change of protocols into these new methods. And one thing that they raved about was the pick list function in the plate reformat software. So the stakeholders that I was partnering with is purely lab bench scientists that doesn't, you know, kind of feel a little bit scared about automation, not sure what to expect, and he was very pleased with how easy it is to use the plate reformat software. So from a UI perspective, he said it was just very friendly, doesn't take a lot of like training or understanding to operate the system effectively. So that was certainly a plus for him. So he talked about using simple CSV file format that everybody, you know, is well accustomed to, and just using this to help kind of shuttle information back and forth was helping him to manage the data portion of it. And even the ability to use you know, Excel to even create the pick list so that he can pre-plan everything before he goes into the lab to conduct the workflow was certainly helpful for him. And one thing that he talked about was the error reports that are generated from the Echo. He was able to use that report to help him create new pick lists so that he doesn't waste any time going into the second round of his studies, and to quickly kind of shift gear if needed once he has data that, that presented to him from the initial studies. So overall, he felt that the Echo is a very easy system to use and highly reliable. And right now, this is really the system in their workflow that they are utilizing day in, day out. And they are actually ramping up in their throughput capability from a few hundreds to tens to 100,000 samples that they are driving towards right now. So I would like to really, you know, thank Maureen and Alex from Labcyte. They not only were great partners, but very good technical experts in helping us, particularly in development of workflow, as well as helping us to design how we should approach our experiments so that it's very effective and that we can do it in a timely manner. And we're still engaged in discussion with them, trying to understand, you know, what other applications are, you know, important to kind of put on our radar system that we could also leverage into building our platform as we move forward. And also, Joby Jenkins from TTP Lab Tech, you know, has been very helpful in this area too when we were doing our assessment, not only from just a support standpoint, but also lending his technical expertise to help us shape the program further.